people recognize earthworms in soil, but they have to have moist soil. That's a real key attribute there. Another thing is that we can see organisms by this fungal mat. It's rare that we can see this, but this has to be really wet conditions. Generally, it's under a canopy of a plant material, and then there's surface material such as these corn residues that are there, and that sometimes occur. So, but the best evidence of microbial activity, of course, are mushrooms. These are the physical structures of fungi that emerge. They have much more mass underneath the soil, but they do emerge and we can see them. But the most numerous organisms in soil are soil bacteria. And we can't see them. We need a microscope. So oftentimes what we'll do is we'll have to quantify them. We put them into a liquid, a sterilized liquid, and then serially dilute it. So in this case, there are many dilutions here. Every time we're taking 1 ml of a sample out and diluting it 10 times. And so by the time you can see only a few colonies, that's when we actually count them. And so we might have 10 to the 9th. So more than 1 billion bacterial colonies in a teaspoon of soil. So soil quality and soil health soil quality was the term originally some 20 years ago, and it emerged to soil health. But they all have fundamental basics in that we're trying to understand the chemistry, the physics of the soil, the physical properties, and the biological properties. All three of these attributes are really important to understand the real soil quality or soil health. And the move from soil quality to soil health really was because we were interested more in biology. There was a hidden recognition that there was more needed to be known about soil biology. And so we looked deeper into the soil biology. And that can be characterized through its activity or its biomass or its diversity. And those three characteristics are very distinct, and they have different ways of telling us about the soil. So these properties and processes that should be related to soil functions, and those functions primarily are very general, but they are things that are important for human life on this planet. And that's decomposing organic matter, cycling water and nutrients, controlling gas emissions, and harboring biodiversity. These functions we could not live without. It's hard to imagine, but we don't think about them hardly at all, but they are extremely important. So there have been many scientists that have proposed a whole range of indicators, and they all can be very useful. But I believe that there are a few key soil health indicators, and they really end up being 4 different categories. And inorganic nutrients are what we basically would take a sample of soil and send it to the NCDA soil testing lab or any private soil testing facility, and they would give you a chemical nutrient analysis for, say, phosphorus and potassium and lime needs. All of those chemical attributes are really important, and we should not abandon that. But there are other tests that are also important and that we haven't been characterizing well enough. And one of them is very importantly, the simple aggregation of soil. It's something that we could do because it's relatively easy to do, but soil testing labs often don't do that, although they're starting to do it now, various private labs. And that aggregation is important for water infiltration in the field. That's really what we're trying to get at, is how much water would actually infiltrate rather than run off. And then there's also soil test biological activity. This is a characteristic that I'm going to explain in more detail through this presentation. And that's characterizing the general activity of soil. And that, of course, is dependent on some of the resources. And some of those resources come from soil organic matter. And that's a biogeochemical attribute that really oversees all 3 of these other ones. So the first step in characterizing biological activity is really to get a good soil sample. And there are many techniques to get a good sample. You can use a spade too. It's done that way. But in our research studies, we often want to use a coring device, because that way we're ensured that we're getting equal representation of the surface sample and anything deeper. But where the biological resources are will determine where we should really sample. So this graphic here shows 3 different land uses across many profiles in Georgia. And you could see that the differences that occur in cropland versus grassland versus forest land really are magnified in the top 4 inches of this profile. And so that if we sample just the top 4 inches, that means that we're going to get the maximum return on our knowledge of what is different about the soil. And if we go a little bit deeper, then we're starting to dilute it with less resource intensive organic matter. And so that can bias the sample. If we take some samples too deep and some samples shallow, we're actually biasing the sample. So we have to be aware of that. So we often start with these cores of sample, we dry them, and then of course they're very coarse. In this case, this happens to be a clay type of soil. And so we want to crush it and sieve it. We pass it through 3/8 of an inch of 3/16 of an inch of a sieve, and we try to get a more uniform. So we're left with the rocks in this case here. And then we have a relatively uniform sample afterwards, because we want to have a homogenized samples, but we don't want to destroy all of the aggregates, because that's where some of the life exists, and we want to reflect what the true biological activity is in the field. So how do we measure soil biological activity, and what does it tell us about the soil? That's the next step of this process here. So this young lady is actually adding water to dried soil. So we have dried soil initially, we add the water, and then the sample on the very left shows the water that has uniformly distributed throughout the sample, whereas the sample in the middle there, or on the right-hand side of that left picture there, it was water, the same amount of water to the same type of soil. It's just that soil was powdered, it was pulverized into a powder, and the water doesn't penetrate nearly as fast. And so we're having an imbalance of the air and the water space there. But we're starting with a dried sample and then re-wetting it up. And that re-wetting process is what we're really trying to capture in a test. We can use field moist soil, but in this test, we want to actually express the biological activity very dramatically through this burst of activity. So these people are putting soil samples into mason jars, similar as if you were pickling your cucumbers or other vegetable products, and we sealed it up with the moist soil and a trap that has alkali in it, and that alkali absorbs the CO2 emitted by the soil. So the CO2 from soil, just like the CO2 from our breath, is a weak acid, and it's absorbed into the alkali, and then we can determine how much CO2 was emitted. And that CO2 is, in this case here, this vision here, is that trap. We add a little color indicator, it turns pink, and then once we titrate with an acid, we can determine how much of that CO2 was in the sample. And that's the way we actually determine it. I thought it would be useful for you to know how we actually determine it. It's a relatively simple setup. Of course, there are a few chemical steps in there, and we have to have controlled conditions in the laboratory. So temperature and water are very important. So these are the numbers that one would get from a test like this, and that basically it comes down to characterizing the cumulative carbon mineralization that occurs over time. So in this case here, we have carbon mineralization as a function of days of incubation. And there are 3 lines projected here. This red line, we have a burst of activity during the first few days, and then it starts to become more linear. And so this becomes stabilized. We would characterize this flatter part as basal respiration. There are 3 depths from the same type of soil here, and it shows that soil depth is really important in how much biological activity occurs. So we characterize this flatter part of the curve with basal respiration, and this flush, which is what we're really interested in the test of biological activity, because we can minimize the amount of time incubating. So that means we can get you a value in the soil test report much quicker rather than several weeks of incubation. So, but the flush and the basal respiration are highly related, and that's the key point here for our analyses. So we can measure at 3 days and at 10 days or at 24 days, and we basically get the same relative difference between these 3 depths. And so that gives us confidence that we're measuring something that's realistic as well as in the field. So you might ask, is the test available commercially? And yes, it is. There are tests that are offered by Solvita or Woods End, and that's the CO2 burst test. And then you can also send it to Cornell, and they have a comprehensive assessment of soil health, and they'll determine that as well. My colleague Rick Haney has developed the test into a one-day procedure. And then in my research lab, the Soil Ecology and Management Lab, we've been doing it for 3 days. And I've been doing it for more than 25 years already. So I think I know what I'm doing, but I want to also point out that every one of these labs determines it a little bit different. And that's why we have a little bit of a dilemma in our science, in that if you wanted to send a sample always to Solvita, then you should always send it to Solvita, because then you're going to get consistency of the result. If you send it only to Cornell, then that's good. But if you send it to one or the other, you're going to get different results because of various processing things. So I process it for 3 days, sieve it coarsely, incubate it, and we're taking 100 grams a sample. We modified that a little bit. We're anywhere from 70 to 100 grams in the future, but we're using more soil than the other labs. And that is maybe not important to you, but it actually gives us more reliability. Another thing is that how we add the water is important too. And so, you could do this test at home, even with simple chemistry that is available to you. You could purchase all these things for a relatively inexpensive amount. It wouldn't be worth your effort, perhaps, but you could do it. And how the water is added is important. This capillary action from the bottom through saturation limits the amount of CO2 that's evolved in some samples, and that's not a good thing. We need to give it an optimal condition of equal water and equal air, and that we could do through 50% water-filled pore space. So the incubation of 3 days is longer than 1 day, and that's a limitation for the method I use. But I'm interested in a good number that I can rely on very much. And so I'm happy to use 3 days rather than 1 day. But the other people are not, so they want to have the analysis in a shorter period of time. And probably the most robust CO2 detection is through infrared gas analysis, as my colleague Rick Haney would do. But all 4 of these methods are reliable. None of them been shown that they are unreliable. Some take more time than others. So that's part of the commercialization. So what we want to know from biological activity is that it relates to other properties of soil. And in this case, I've related soil test biological activity from some samples that were taken in Georgia and related it to mean weight diameter of water stable aggregate. So this is the aggregation portion of the soil. And we see that there's a relationship here. There's a strong association. And so if we wanted to know about aggregation and about water infiltration, we would get a little bit of that information just by knowing soil test biological activity. Equally well, we'd related here biological activity in Kansas and in Georgia, 2 different locations, 2 very different soil types. One is an ultisol and one is a mollisol, and related that to soil bacterial diversity. And there was it was weaker association, but it was equally determinant in that there is an association. And of course, I've dropped out one point here from the Kansas location, but in general, it's amazing that we actually found pretty good association here, because it's not something that we would expect, that activity is absolutely associated with diversity. But in this case, the samples were highly associated. And then finally, one thing that I think the test of biological activity is really showing us is that its association with soil nitrogen mineralization. So in this case, again, we have biological activity here on the horizontal axis and net nitrogen mineralization on the vertical axis. And we can see that there's a strong association on soils taken from North Carolina and Virginia that are relatively sandy, in this case, less than 20% clay. And then if we plotted those soils that had medium texture, where they had some more sand more clay and a little bit less sand, and then also soils that had high amounts of clay, so perhaps more from the Piedmont and the Shenandoah Valley in Virginia, that where samples had higher clay content, but all 3 of these relationships were essentially the same. And so these are the same data, just plotted here on same without any distinction of where they came from. So there's one association that occurred. And so soil texture is important in determining biological activity, but the relationship between biological activity and nitrogen mineralization was not affected by texture. So how could we use this? If we wanted to know how much nitrogen is mineralized in soil, and nitrogen mineralized in soil is an important attribute, because if you took a soil sample and send it to the NCDA soil testing lab, they could tell you how much inorganic nitrogen was there, but they wouldn't tell you how much organic nitrogen or mineralizable organic nitrogen was there. And that organic nitrogen mineralization is important. It's important for, say, organic producers, because that's what they rely on, organic substrates that have to be mineralized, have to be decomposed by microorganisms. It's important for conventional row crop farmers as well, because we want to know how much of the organic matter is being mineralized. So in this case, if we had a soil test biological activity of 200, we could say that it has 55 pounds of nitrogen per acre in a growing season. And so that would be useful to know. But if we had 500 parts per million of biological activity, we could say that the soil had 127 pounds of nitrogen per acre in a growing season. And so that would be quite a dramatically different situation. So we might want to know that, because nitrogen fertilizer, first of all, is not something we need to apply abundantly, because it leaks in the environment. And also it costs something, and it's costs a lot more these days than it did before. So this association between nitrogen mineralization and biological activity was explored through some field studies. So I conducted various field studies, 47 trials in 11 counties in North Carolina and Virginia. And what was found, and I know this is a little bit complicated, but I'm going to try to be deliberate about this. This is the corn grain yield response to initial dose of nitrogen fertilizer. And I'm going to explain that a little bit more in the following graphs. But it's essentially the pounds of grain of corn grain that's produced for every pound of nitrogen that is applied. And surprisingly, well, it wasn't surprising to me, because I was kind of expecting these results, but it just validates the approach that soil test biological activity, the greater the biological activity, the less responsive the plant was to nitrogen fertilizer, meaning that soils had an ability to supply different amounts of nitrogen to the plant from organic nitrogen mineralization. So I'm going to show that the values on the left side of this graph had high grain yield response to nitrogen fertilizer. And that would be characterized in a typical response curve, where we have apply an initial dose of nitrogen fertilizer and we get this very large increase in yield. Now, these are cases where the soil is just not supplying enough nitrogen, and by having applied nitrogen fertilizer, we get a very strong yield response. And that's basically what cooperative extension is telling us, is that if you have a corn plant, you need to fertilize it. But there are cases in the middle here where the yield response would be, with that same dose of nitrogen fertilizer, would be much less. And then further, on the far end of these data points here, is that we have a very flat curve where the corn grain yield is not responding hardly at all to additional nitrogen fertilizer. And you might say, well, that's a poor experiment. But actually it's not. It just means that there's an abundant supply of nitrogen in soil from the biological activity. So we would want to know that and know how much for nitrogen fertilizer to apply. So the data were then summarized in this graphic here. So this is the nitrogen factor for economically optimum production. This is the pounds of nitrogen per bushel of grain. And in general, if you've ever heard of, and maybe some of you are corn farmers, and you might have heard of field corn where you apply 1 to 1.2 pounds of nitrogen per bushel of grain. So right at this point here. And currently, that's kind of the recommendation. It's approximately 0.9 to 1. It's in this range here, 0.9 to 1.2. That's how much you might be told that's how, if you wanted to grow 100 bushel of corn grain in the field, then you'd apply 100 pounds of nitrogen, essentially, give or take a little bit. And that works for soils that are low in biological activity. But those soils that had high biological activity, they didn't need near as much of that nitrogen fertilizer. So we've now changed the paradigm from one of a standard rate of nitrogen to one that could actually be variable depending on biological activity. So this range here would be the normal range, which would be recommended, but we're changing that. So the final question that I have, and it goes for a little while yet, I got plenty of graphs to back this up, but how does one change soil biological activity? Well, we conducted these nitrogen trials on corn, and we conducted 99 field trials over several growing seasons. So we conducted more than just the first 47. So we had 10 of them that were conventional tillage and 89 of them that were under no tillage. So I was trying to target producers that were using more of a conservation approach. And so the biological activity in those fields that had no tillage or conservation tillage was statistically greater than those fields that had conventional tillage. So that gave us already some interesting results that biological activity is greater when we use no tillage. That's in the top 4 inches of depth. And then nitrogen mineralization, on average of these 89 fields versus 10 fields, it was also greater under no tillage than under conventional tillage. So same correspondence as biological activity. Corn grain yield, in fact, at the optimal nitrogen level, was also significantly greater under no tillage than conventional tillage. And the return over the standard rate of nitrogen recommendation was significant, suggesting that actually, in this case, this is the return. We were getting greater economic return under no tillage than under conventional tillage because of the biological activity that reduced nitrogen mineralization, or that reduced the need for nitrogen fertilizer because of the high nitrogen mineralization. So we also asked the question and sorted the data by animal manure amendment. So if we had soils with no organic amendment and those soils with organic amendment. So we had 72 without and 27 fields with. And the biological activity was greater with organic amendment, something that we would expect because that organic amendment is supplying organic substrates. The nitrogen mineralization was higher as well. And the corn grain yield was no different in these two comparisons. But the return, the profitability over the standard nitrogen recommendation was greater with organic amendment because we were substituting some of that organic amendment, that nitrogen fertilizer, by the organic amendment. And in this case, I wasn't accounting for the cost of the organic amendment. This is just the difference in the profitability over the standard nitrogen recommendation, on that metric simply only. And then we had a comparison between cover cropping. We had some fields that were no cover crop, some with single species cover crop, and some with multi-species cover crop. Biological activity tended to be greater with multi-species cover crop. So there was greater carbon sources from that plant material, that diversity of plant material that was added to the soil. And nitrogen mineralization was also greater with the multi-species cover crop. And corn grain yield tended to be greater with any other cover crops than with no cover crop. So we can protect our soil and enrich the soil by using cover crops. And here we see some variability with the economic return over the standard nitrogen recommendation, but it was always better. It was better with the multi-species cover crop because we were reducing the nitrogen fertilizer that was needed because we had greater nitrogen mineralization. And the biological activity indicated that reduced nitrogen fertilizer requirement. So I also conducted some trials in a broader geographic region, from anywhere from Georgia up to West Virginia, and including most of the sites in North Carolina, where we had tall fescue. And tall fescue is a common forage throughout this region, and it is often stockpiled, at least for those progressive growers. They're stockpiling their fescue, and we asked the question, could we actually reduce nitrogen fertilizer? Because the standard recommendation is to apply anywhere from 50 to 100 pounds of nitrogen per acre on stockpiled tall fescue. And so it was a convenient approach for us because the forage is easy enough to handle, to harvest. You just have to mow grass in December and January, and then we can get these data. So we had 92 fields sampled over 3 different years. We had a comparison between conventional and improved forage utilization. So that would be conventional would be general stocking that was continuous, and improved was a variety of things, but it generally meant that rotational grazing was part of that. In pasture age, we had anywhere from less than 10-year-old pastures or to greater than 40 years old. And so we characterized them in different categories there. And then we had elevation from anywhere from the coastal plain to the Piedmont to the Appalachian region. And soil texture varied from coarse to medium to fine. And so we had these different categories, and we separated out some of the data. And I'm going to share you just a few of these categorizations. So these are the soil properties in the top 4 inches as affected by forage utilization. And you might notice that in the corn studies and in the tall fescue studies, I've used 0 to 4 inch depth. And I believe that that's a very appropriate sampling depth for conservation management systems. Now, I believe that the current recommendation is still 0 to 6 inches for any conventional type of management. So we had conventional management in this case, continuous stocking, and oftentimes haying of the pasture as well. Whereas we had improved management, and that oftentimes didn't include haying, and it oftentimes did include rotational grazing. But there were some other characteristics there too, but those are the kind of the broad categorizations. So it's not a typical experiment, but it's a survey of what happened on these 92 fields. So total organic carbon was greater under improved management than conventional management. So that's an observation that is important to understand. And particulate organic carbon, this is the readily visible organic matter that occurs when we sieve the soil under a wet sieving, and we can actually see some of the organic particles. And that was greater as well. So that oftentimes reflects roots and surface residues. The cation exchange capacity was elevated under improved management than conventional management because part of that is because of organic matter. The higher the organic carbon, in this case, total organic carbon, it really implies total organic matter. And that higher organic matter gives higher cation exchange capacity. And the sieved soil density was lower under improved management than conventional. That means the soil is fluffier, it has more organic matter, and that organic matter is lighter, and so the density is lower when we have higher organic matter. The forage actually was in a better crude protein content under improved management, and the dry matter response to nitrogen fertilizer was reduced under improved management because the higher organic matter, higher biological activity led to a less of a need for nitrogen fertilizer. So the dry matter response was muted under the improved management. So here's a graph that I wanted to put in your mind that I like numbers. So I like graphs. You may have a hard time with my presentation because of all the graphs, but if you have questions, I'll try to answer them. But in this case, I'm plotting soil organic matter against pasture age. And I want to put it in your mind what you think might happen with this relationship. I'm going to put up also total soil nitrogen with pasture age. And basically, I'm going to explain that the responses should be really similar because soil organic matter, when we measure total carbon, is highly related to total soil nitrogen. And these are the data that we observed. We had pastures that were young, we had the lowest organic matter. And that's because they typically were in a cropping rotation prior to the pasture. And so once we had the pasture for more than 10, 20 years, after 20 years, it seems that there was more of a stabilization. So we could actually calculate the carbon sequestration rate from a pasture establishment period. For the nitrogen, there was less of a plateau, but there still was a plateau, and it showed the same response, that there was an increase in total nitrogen with pasture age. So obviously the farmers are applying some nitrogen to the young pastures, but at some point that pasture has become mature enough that nitrogen is no longer needed because it's recycling the nitrogen. So there's one other graphic that I wanted to point out to you, and that I want you to think about, well, how could soil test biological activity relate to soil texture index? So in this case, I've created an index of the clay divided by the sand, but it basically, from moving from left to right, we have greater clay content. So we might say that in general, we might be moving, say, from the coastal plain on the left to the Piedmont on the right, or a mixture, because all 3 of the physiographic regions have different soil textures. But in general, the coastal plain has sandier soils than the Piedmont. But the soil test biological activity was low, with soil texture index that was low, and then it increased and then plateaued. And so it shows also that soil texture does have an impact on soil test biological activity, but the association between biological activity and nitrogen mineralization is independent of texture. So these data show that texture is important, but it's not on the calibration with nitrogen mineralization. So here are 2 sets of plants. My daughter actually grew it for a science experiment. but I thought it was very useful for us to envision. So what's different from the plants, the group of plants on the left versus those on the right? You might see that some of the plants, well, most of the plants on the left are shorter than the plants on the right. The plants on the left tend to have maybe generally fewer leaves than those on the right. The leaves are greener on the right than they are on the left. And so these are characteristics of nitrogen deficiency on the left compared to nitrogen availability on the right. And in this case, none of these plants were fertilized with inorganic nitrogen. So there was a difference. And of course, I'm going to try to explain how that difference might have come about. If we have no nitrogen in the soil, no available nitrogen from whatever source, we have no yield. It's as simple as that. You go to the beach and grab some sand that has very little nitrogen, and you plant something in it, you get nothing out of it. So the available nitrogen is important for yield. There's no doubt about it. But we turn the page, and available nitrogen also leads to high amount of environmental degradation throughout the world. All of these coastal estuary regions around the world are being polluted, especially where countries use a lot of industrial nitrogen. And that's occurring all around the world. And it's something that we have to get better handle on, because we cannot afford to pollute our environment, because we live in this environment and we are going to be affected by it. So this idealized response to nitrogen, I wanted to characterize that a little bit further. So the field in the background are some of the tall fescue trials that we conducted. The available nitrogen can be characterized as inorganic nitrogen in the surface or deep in the profile. Organic nitrogen can be long-term stable material, or it can be biologically active. And of course, you might think that, well, available nitrogen also comes from the fertilizer in the bag, or fertilizer in the truck, or in the backpack sprayer. That source of nitrogen also is external, but these are the internal sources of available nitrogen in soil. So those sites that have low nitrogen availability will have high nitrogen fertilizer response, because they just don't simply have enough nitrogen to produce the proteins that they need to produce a robust plant. And so in those cases, we need to apply fertilizer. How that's applied, the source of that is up for you to decide. And then if we do that, we're going to increase farm profitability or enterprise profitability. That's an essential point. We need to apply nitrogen to get better plant growth in some cases. But there are cases where the soil has enough available nitrogen, and if you are using organic amendments and supplying that soil with available nitrogen, then maybe you have to consider slowing down, because you can have too much of the nitrogen. And if we have that high available nitrogen, we want to reduce the environmental impact by not applying any more nitrogen. And that's important. So in general, we want to shift from the left side to the right side. And if we can do that, that means that our soils are healthier, because they have more nitrogen in them to supply the plants with nitrogen. But we want to do that without causing too much nitrogen leakage. So my research is really focused on surface soil inorganic nitrogen and biologically active nitrogen. So can we predict soil organic nitrogen mineralization? Well, yes, I've shown that to you already, and we can do that. And these graphics here show that the more soil test biological activity we have, the more nitrogen is going to be mineralized. It's a proven fact, because we've done this many times over, but there are complications. We can have too much biological activity, and then all of a sudden we have nitrogen immobilization, and that can occur. But in general, for the thousands of samples I've analyzed, there are very few of them that actually lead to net nitrogen immobilization. So the magnitude of forage yield response to nitrogen fertilizer input and economically optimum nitrogen rate would decline with increasing net nitrogen mineralization in soil test biological activity. That's the hypothesis that was driving this tall fescue research. And I wanted to give you these examples because it illustrates some points. So the relative yield that we would expect when we add no nitrogen fertilizer, the hypothesis in the beginning was that if we didn't add nitrogen fertilizer, we'd get very low yield. And then if we added fertilizer, like the recommended rate of 100 pounds of nitrogen per acre, we'd achieve more of the target optimal yield that we were looking for. On the other hand, if we added no nitrogen fertilizer and we got near optimal relative yield as compared to adding fertilizer, then that meant that we had high nitrogen mineralization and we didn't need to apply nitrogen fertilizer. But without a test, we wouldn't know. How would we know? Because we didn't have a test before. So that was part of this development of the soil test biological activity. So again, we stockpiled these trials and throughout the eastern U.S. So we collected soil. This just happens to be an example of a soil probe where we're cutting out the soil from the 4-inch depth. And then we're applying the different nitrogen fertilizer rates to small plots in the field so that we can characterize with accuracy the amount of yield response to the nitrogen fertilizer. We'd record the weight of the forage. We had to collect it. We had to collect out of every field. We collected, I think, 32 samples. So we were collecting both above ground material as well as plant material close to the ground to really characterize what was the yield response to the nitrogen fertilizer. We had 4 different nitrogen fertilizer rates. And then we had we set different thresholds for this cost of value condition. So the fertilizer costs something and the forage that was produced has a value. And so we can shift that cost of value threshold. And that's shown here on the right side here. This would be a low cost of nitrogen fertilizer. This would be a medium cost of nitrogen fertilizer and a high cost of nitrogen fertilizer. But it's not just the cost of fertilizer, it's also the value of the forage. And so we make that ratio, and that's how we can get 2 different numbers with 2 different sets of conditions here in the middle. Well, when we look across sites, and I'm almost done here, but I wanted to summarize what happened. So in this case here, this is the statistical distribution of the samples under the corn trials that I conducted and the tall fescue trials. First of all, you'll see that there was, in general, greater biological activity when we had tall fescue on the field rather than corn. Now, these were different fields, of course, but in general, we sampled so many different fields, 158 versus 92 of them throughout a large region, that the implication is that perennial grass leads to greater soil test biological activity than corn. And that's a general statement, because obviously we can have a field here that has relatively low biological activity compared to a cornfield that has high biological activity. But in general, there's a shift that occurred. So basically on this distribution, I've created 5 different categories of biological activity: very low, low, medium, high, and very high. And this basically then leads us to how we could develop a nitrogen recommendation based on biological activity. And these are the points here. The 5 points are, this is the very low, the medium, the high, and the very high, the average of those points. And we fit the relative yield of the tall fescue to soil test biological activity. And it basically showed the ideal nitrogen response. It showed that, of course, we're extrapolating in this case here, but it basically led to no yield with no biological activity. And then it increased dramatically and then tended to plateau once we got to a certain level. So that means that maybe there's an optimal level that we should be shooting for, and not necessarily endless activity that we would never achieve. So essentially, we can then characterize that as these categories for soil test biological activity, and that could give us an idea of what to expect. If we didn't fertilize at all, what kind of yield could we achieve? And if we didn't fertilize in a soil that had high biological activity, would we even really need nitrogen fertilizer at all? And these are the data where it shows the economically optimum nitrogen rate. So we would have in these fields that had low biological activity up to 200 pounds of nitrogen per acre. The current recommendation is for 50 to 100 pounds of nitrogen. And so that means that those fields that have very low biological activity are kind of in the range of what the current recommendations are. But whenever we went up with higher sites with higher biological activity, we needed much less nitrogen fertilizer. It was never zero, but that's because we had a diversity of sites. And of course, you have to take some chances. And in this case, this is all statistical distribution. That's how recommendations are made. And if we put this further into the nitrogen factor, this is now scaling the pounds of nitrogen per ton of forage. And so it takes away from the fact that some sites might have just relatively poorer soil because of water limitations, because of acidity, various other limitations, but this puts it on an equal basis per ton of forage. And then this other graphic, and I'm putting them together here, shows the difference between a low cost value threshold of the nitrogen, the fertilizer nitrogen to the value of the forage. And we could have then two different curves there, and that gives us a range of recommendation that we might be able to achieve. And in this case of the biological activity, if fertilizer cost is high, there's really no good reason to apply nitrogen fertilizer above a level of about 200 parts per million of biological activity. So this gives us really strong evidence of how we could actually change our nitrogen recommendations to increase profitability as well as reduce environmental degradation. So in summary, soil health is a vital underpinning of sustainable agriculture. Soil test biological activity is a simple, rapid, and robust indicator of soil nitrogen availability. Productivity and environmental quality are strongly linked through soil health assessment using soil test biological activity. And so moving this standard approach where we have, say, 1 pound of nitrogen per bushel of grain for corn grain, or for a unit of nitrogen per ton of forage, that, it's a relative basis here. That standard approach where we would know nothing about the soil and you just go to the recommendation book and says, okay, which crop, this is the amount of nitrogen you have, you'd have a standard approach. You'd apply 1. We're now moving to a soil health approach. We should be moving to a soil health approach. The data that I've presented would suggest that we should, and that we actually have higher biological activity, which is good because that's what you as a grower want. You want better soil. Better soil means higher biological activity up to an optimum, maybe not excessive, but up to an optimum level. And then we could actually, in this case, we would have applied less than adequate nitrogen if we had soils that were really poor. But we would also apply too much nitrogen in many soils that had a high biological activity. So this paradigm is shifting the way we think. So improving soil test biological activity over time is an indication of greater functioning of soil. And that functioning of soil, we can have measurably greater supply of nitrogen and possibly other nutrients. We haven't explored all the possibilities yet. And we likely have more diverse soil microbial community structure. And so that's good for the functioning of the soil. We likely have greater water infiltration and storage in soil for plant uptake because we've shown you the relationship between aggregation and soil test biological activity. And we can see that in soils that are in good condition, that have high biological activity, they allow water to penetrate the soil profile. And almost certainly greater soil organic carbon storage and possibly reduced total greenhouse gas emissions if we have high biological activity in soil. And certainly we might have the potential resilience to biotic and environmental stresses. And that's our challenge, is to improve our soil for the benefit of us as well as the global community. So if you have any questions afterwards, without the chat, you can email me here at this location here. And at that point, I am ready to take any questions that you might have.
Thank you very much, Alan. That was wonderful. Currently, the only question I actually answered one of them. Oh, that one just came up. Okay. Would a nitrogen recommendation from your lab come out about the same as, say, from Cornell?
Depends on how about is close. So the Cornell does their test for 4 days, and most people would think, well, I do the test for 3 days and Cornell does it 4 days. They should get a higher value from Cornell because the longer you incubate soil, the more CO2 is going to have. It's cumulative. But in fact, when we did the comparison, we actually got less from Cornell because there are all these technical difficulties, because there are at Cornell, they incubate the soil at a lower temperature. So that means that biological activity cannot be higher at the same incubation period. And so, but in general, yes, if you sent your sample, your soil, say, over time to the Cornell, I would recommend doing that continually. It's not good at this point, since we don't have the calibrations, to switch among labs.
Yeah, it seems like that's kind of for almost all measures that you have a high degree of dynamic nature to soil, even depending on like what time you sample the soils, what time of year and things of that nature, that I think sticking to the same kind of similar pattern all the time, then you have more likely ability to compare, possibly.
Yeah, of course. I think that that's a good strategy, you know, and Janelle, you've got a, you know, a research background, and maybe others do too, but those details are, you know, the finer details. I think in general, if you sent your sample to Cornell, it would come out in a general sense, compared to your neighbors, let's say, uh, however that is, better or worse, and you send it to my lab, in general, it would be about the same, let's say.
Yeah, I mean, it's a good measurement to have regardless of who you send it to have some measure of soil biological or not. I mean, you know.
I think so, because especially when you do it more than once, then you'll start finding out, well, what did you do to the soil and how did it respond?
Yeah, that's a really good point. Oh, we have one more question here. How would you realistically increase biological activity in a year-round row cropping system? Would it be feasible? And could you increase it enough to make a difference in nitrogen availability?
Great question. So the way to increase biological activity is through organic resources. So you could add compost. That would be the quickest way, I believe, to change biological activity. It may not immediately change nitrogen availability because the compost is oftentimes high carbon to nitrogen ratio. It possibly would not mineralize immediately. That's a factor that has to be analysis of the compost itself. But over time, say over several years, over three, let's just put a time frame of 3 years. Over 3 years, I believe that yes, it would increase nitrogen availability and reduce ability to reduce nitrogen fertilizer because of that. So there are other ways. We don't want to disturb the soil. And when we disturb the soil, we oxidize these and we enrich the biological activity in the field too much at one time. And then they don't have any resources. And that's an important aspect. That's why conservation tillage works in the long term, is because we're conserving those organic resources rather than allowing the organisms to chew them up and deplete them. That's what our conventional agriculture has done. It's exposed all the organic resources and there's nothing left.
Mm. So would you suggest in that way, if they're looking at making a change within a year, that maybe perhaps a combination of, say, a leguminous cover crop or mixed, that you could maybe destroy in a way that you're not tilling it, and then add compost, you know, also, or a combination of something like that to be able to, because I would think the legumes would, when they break down, have the ability to really set nitrogen a little bit sooner, or is that not the case?
Absolutely, sure. Combinations are always best. I mean, I think that's the balance, you know, of nature. We want to have combinations. There's never one solution. So adding legumes in the rotation with the compost is great. And of course, there are certain legumes that are even are typically better than others. If you've ever watched hairy vetch decompose, you can almost see it in front of your eyes how it decomposes from one day to the next. It actually turns black very, very soon with precipitation and dew in the morning. It decomposes very fast compared to other legumes that you know, like soybean stems. They don't decompose, but it's a legume, but they don't decompose very fast. Anyway, so those combinations are really important. Absolutely.
Then there's the other question: does nitrogen availability vary from sandy to clay soils, meaning the texture influencing that?
Yeah, the data that I've collected would suggest that the sandier soils have a harder time increasing nitrogen mineralization, because the organic resources are just not able to persist in the soil. So in general, that doesn't mean that any specific sandy soil can't have high nitrogen mineralization. It can, but in general, sandy soils have less nitrogen mineralization potential than clay soils.
Yeah, it's kind of a bummer for those on the coast. So I just launched the poll. And again, I want to thank you very much for your time. This is a really informative presentation. For those of you who haven't seen, these are a monthly occurrence. I have them scheduled all the way out until November. Actually, I will put in the chat the link to all this information of what is coming up. Next month, yours truly, myself, will be presenting on how to deal with soil health in a high tunnel, because when you're placing some cover over your soil and you're not having natural rainfall, and you can have a little bit of a different situation. Sometimes there's some salt buildup with fertilizers and things within that. So I'm going to discuss kind of how to manage your soil health within a system that is not outside, essentially.